Thank you, Dr. Melnikov, for that really very informative lecture. The next lecture will deal with opportunities in industry. I want to say that we're still waiting. I'm still hoping that Dr. Budol makes it. He's expected to come. I hope he didn't try to board an aircraft with his gun or something like that, but I also hope he's all right because I fully expected him to be here. But the next presentation will be by Dr. Steenbergen from Cubist Pharmaceuticals, and she's going to tell us about microbiology and industry. Judy?
All right. Hello. I'm glad I'll be talking about the microbiology and industry. I'm only going to be discussing my perspective from what I've done and from what I know from my company, since it's the only industry company I've worked at. I work at Cubist Pharmaceuticals Incorporated. It's in Lexington, Massachusetts, just outside of Boston, where the BIO conference was recently. It's a fully integrated pharmaceutical company. It's an entirely anti-infective company. We have a transition from a biotech company, early startup, to a pharmaceutical company when FDA approved our only approved antibiotic at this point, daptomycin, in 2003. I have about 400 employees now, and we have all the different components of a larger company. We have research and development, takes up a large portion of the company. We have a large commercial base, and all the other components that go into a company. We have regulatory, manufacturing, quality control, and senior executives. This is the antibiotic that we research, and just a brief background, because I'll be discussing what my interactions have been with daptomycin. Daptomycin is a lipopeptide. It is a natural product produced by Streptomyces roseosporus. It has gram-positive potency, and it was FDA approved for complicated skin and skin structure infections in 2003, and much more recently for Staph aureus bacteremia and right-sided endocarditis, and that just recently happened in 2006. This is the overview of the development of daptomycin, and I'm sure most of you have heard, and it keeps going around, how long it takes to get an antibiotic to market. We actually in-licensed the product from Eli Lilly. They had shelved it due to toxicity issues, and we were able to revive it. So we got the worldwide rights licensed in 1997. And then within a year, we worked on a different dosing regimen to avoid toxicity, and were able to file additional patents and filed our investigational new drug application, the IND. Once that's filed, we were able to put it into people, and we started our clinical development phase of daptomycin. The package that was submitted to the FDA, the new drug application, was filed in late 2002, and we got final approval in 2003. After that, we launched the drug for commercial availability. And here the box outlines when I started at the company. I started after the approvals, after most of the discovery work had been done, after the clinical trials had started. So what was my role with daptomycin as a microbiologist? So my job, I manage the portfolio of external investigator studies, and I'll go into much more detail on that in a minute. I present scientific data externally at advisory boards, at company meetings. I coordinate with our regulatory group to help file any regulatory documents that need to go out that have microbiology information in them. There are other commitments that the FDA puts on us and requires us to do, and if they're microbiologically based, I write those, I get those ready for filing and get them through the correct processes for the FDA. I also manage external studies with vendors or contract labs to get additional studies that we need for both the FDA requirements, for our own knowledge, and I manage those. I manage quality assurance and control of microbiology data that does come in, making sure what comes in and what goes out are accurate and that all the correct controls have been run. And I provide scientific support for marketing initiatives. Since we do have an antibiotic, there's a large component that is microbiologically based. So when marketing puts pieces together and they want to incorporate anything that has microbiology components, time kills, susceptibilities, I'm consulted and we work together to get the most detailed yet simple graphics that we can to illustrate this type of data. I also support various medical affairs groups, support our global partners, and I also am the lead microbiologist on ongoing clinical trials at Cubist. So, first, the external investigator portfolio. This is why I was hired at Cubist. This was the original responsibility and majority of the work I did the first year and a half, 2 years that I was there. These studies are research and development sponsored external studies to support ongoing Cubist initiatives. They are studies that either come in through external investigators that are not initiated by us. They have an interest or they have a model system that they're interested in using our antibiotic in. We're the sole holders of the antibiotic. They'll contact us with a full protocol and ask questions. I'll work with them, discuss the protocol, figure out if there's things that they're missing, there's things that we're interested in, to come up with a mutually beneficial type of protocol. One, so they can continue to do their academic research, and two, that we can get the types of publications at the end of the day that we want to see as well. I coordinate an internal review group that we review all protocols that come in. I communicate the comments back to the primary investigators. We work again to make sure that this is up to what we want. And then we go through a lot of what was discussed before. We do contracts. We do material transfer agreements. We determine if we see any potential IP or intellectual property issues with any of these studies, and we try to, again, control our assets in that as well. And believe me, all academic institutions have their lawyers looking at everything and pushing back on just about everything as well. Worry about the budget, making sure it fits in with our clinical microbiology group budget, and then the study can finally get initiated. I can have powder sent, and they can go on with their work. I then also track the study, expected timelines, when do they hope to get this done, and I do regular follow-up. There can be a lot of glitches that occur. For something as simple as susceptibility testing, daptomycin, the antibiotic that we work with, has a calcium dependency, which is not an issue physiologically because calcium is present. However, in some media, there's not enough calcium. If they weren't aware of that prior to starting, they may get some aberrant results. And then it's my job, and one of the reasons I contact them regularly, see how things are going, is to avoid them having done a huge amount of work, a huge experiment, animal modeling, and the in vitro data doesn't line up. So just to get all of the communication out in the open. Then I encourage them to present. I encourage abstracts, posters, and full publications. The reason we do these studies is to get that share of voice out there. We want our antibiotic, if you type into Medline, our daptomycin, we want articles to pop up, and we want recent articles to pop up. So for me, I still push on the publications, and everything that comes out of our studies, I do review all those publications prior to them going out. So what do you need to know for this portion of my job? Well, you need to know experimental procedure and understand the protocols. You need to keep up with current literature. This is exactly what we've always done as far as scientists go. You need to understand it, you don't get it, you read about it, and you make sure that the things that are out there are included. If there's a new, interesting virulence factor that all the hot literature is talking about, you want that included in the next study that some investigator brings to you, and you want to have that discussion with them so that you can have an intelligent conversation and say, oh, well, this is the method that I've read about that seems to be the way people are looking at it, and they might communicate and say, oh, well, we're working on it this way. It's great, show me those results and we'll move that forward. So you just need to stay on top of it for both your education as well as making sure that whatever does get funded and does move forward is the best possible study that could move forward. I also need to know what the company goals are so that you can integrate the correct studies at an early time. A lot of this comes down to commercial and marketing initiatives. And if marketing says we want to expand, we want to look at we want to expand and market to oncologists this year. And well, clinical data is tough to get. To get human trial data, to get any kind of case series, any of that takes a lot more time, a lot more approvals, and a lot more money. But microbiological data is faster, easier, and the turnaround to your first poster, your abstract, anything that can be cited and can help in this initiative is faster. So as soon as I know what the goals are for that year from our marketing group, from any of the groups, I start approaching individuals. So if as I mentioned, if I was going after oncology, I would contact some of our microbiologists that we know at some of the major cancer institutes in the U.S. and say, hey, do you have an idea for a study? You still want them to worry about what they're doing. You don't want them to go outside of what they're good at, and you want them to be excited and interested in their projects too. We're not telling them what to do. We want them to tell us what they think we need them to do. And so working with them, whether it's kill curves or getting some additional data out there that we can then move forward for our marketing to get this product and continue to sell it. So then you also need to be able to interpret and discuss the data. As I said, if some data comes back and it's unexpected, why? What happened? And it's the same as any of us who do the lab research and do the bench research. Something goes wrong. It's best to figure out why. And whatever that answer is acceptable. It's just let's understand it because we don't want to be surprised in the long run. Interpersonal interactions are key. I communicate with these investigators very regularly. The communication has to be there. There has to be trust. There has to be understanding. And they need to realize that you know what you're talking about so that you can continue to have these discussions and that they're willing to come to you. I have many investigators that'll pick up the phone and call me at the slightest issue they're running into because they do know that I know the drug better than they do. And they want to learn everything they can about that drug. So you also need to be able to convey the resulting data to a variety of groups, from low, medium to high amounts of data, to process it and get it into a form that they can understand. And there's obviously organization. At any given time, I'm coordinating 45 to 65 of these external studies. I need to know who's doing what, who's expected to publish when should I expect to see those posters before they print them, and I need to understand when we're hitting some kind of hiccup, and also to prepare our other groups that this type of data is coming out. There are negative studies that result in negative findings. Be prepared that this is coming out, whatever that means. So just to keep all of that organized and running. I drew a quick diagram just to show you again where all of these external studies came from. First, there's FDA commitments. When a drug is approved, the FDA hands down a list of things they want you to continue to do or the requirements you're required to do. This includes annual reports that have to be written and sent to them that contain any new interesting animal data, any new large groups of strains that you've looked at that are unique, and to surveillance studies. Any antibiotic that gets approved is required to continue to look at wild-type strains of the approved bacteria that we're approved for to make sure that the drug stays susceptible and what percentage are susceptible out there. These are FDA commitments. So those types of studies, I know that this is the FDA requirement, and I specifically go look for individuals who can do these studies, that have the correct good laboratory practice procedures in place, that know what they're doing, and I can get this data to the FDA in a timely manner. Others are Cubist-initiated, and these are studies where if we're interested in elucidating the mechanism of action further or the mechanism of resistance, or if there's a new animal model that we think would be interesting as a look into whether or not we want to look at that type of disease clinically. So we'll initiate those and we'll find the individuals in the field that we know are that have that model that is unique. And then finally are those that we don't look for at all and that just show up. They send them to me and we route those all through this particular procedure. And our goal at the end of the day is at the bottom of this, where we want publications. Like I said, we want to share a voice. We want our drug to have as many publications as the other drugs that are in the same anti-MRSA range that we are. This is primarily for commercial purposes and also to inform investigators and individuals out there about our antibiotic and that we are continuing and having continued interest in what's going on with our antibiotic. For the modeling type work, we're interested in that for drug discovery purposes and for clinical development. As I mentioned, if there's a new animal model looking at some clinical disease, that we can then present that to our clinical development group and say, this is some interesting data, and they can choose what to do with that. And then the last area is our key opinion leaders. We have a lot of individuals that are key in the field of infectious disease, and we need to keep them engaged. A lot of them do not necessarily want to have communications with our commercial group, so to be able to have scientific interactions with them has been very helpful to getting them to understand more about the antibiotic and learning about what we do have, and also to get them to do their research and to put their name on something with our antibiotic, which is beneficial both clinically and commercially. So as I mentioned, when I started, that was my primary job. I still do all of that, and now I'm incredibly excited that I'm also working on clinical trials. Since we do have an antibiotic, we have antibiotic clinical trials, and with antibiotics, there's microbiology involved. Therefore, this is unique to the antibiotic world. The microbiologist is involved throughout the study, and I tried to highlight it here, and I think I have a mouse somewhere. Nope. Oh, yep. Okay. So from the concept, anyone can come up with a concept within, at least our company, again, we do have a small company, come up with a concept for a new clinical trial. At that point, it's decided, is there going to be a microbiology endpoint? Is microbiology important in this specific trial or not? If it is, the microbiologist, in this case, me, is included in the discussion as far as concept. What organisms are we looking at? Are we going to only look at Staph aureus? Do we want to look at Enterococci as well? What types of gram-positive pathogens should be in this particular study? We have clinicians involved. They're determining what the primary causative agents are in the clinical stage. Then we put together protocol synopsis. This is an incredibly brief outline of everything that's going to be contained in this trial. It includes your exclusion criteria, your inclusion criteria. It will determine outcome, what is defined as a microbiological success, what is the baseline pathogen, what is a superinfecting pathogen, what is defined as a failure. And as the microbiologist, you're responsible for knowing all of those things with the organisms you're looking at, what time points within that study you're looking at failure, and what microbiological failure looks like or what microbiological success looks like. Then there's a point within all of this where there's feasibility. Are we going to be able to get the patients we need? And there's a microbiological component to that as well. What are the MRSA rates? If we only want to include MRSA in a trial, how are we going to do that trial? What are the rates? Where are the rates higher in the US, in Europe? Where can we find these patients that we're looking for? So there's a feasibility component involved. We then do the full protocol development, and everything's expanded, just per the FDA guidance, exactly what they need in each of the sections. And there is a large microbiology section, including some preclinical data, what led you to making this decision to move forward. And those would be those animal models I discussed that I work with in the external investigator studies, and incorporating all of that into this protocol development. You submit it to the FDA, you wait to hear back, and then you can move forward, depending on which type of study this is. There's all sorts of phase 1, phase 2, phase 3, phase 4 types of study, and all of this will be dependent on that. I'm just doing the overview. There's a site initiation or investigator meetings where I go to and I present the microbiology. Our clinician will present the clinical points of it, and it's to get everyone on board who will be a site for the clinical trial. And then the trial runs, and depending on that, how long that takes and what the enrollment is. But throughout that, there are other things that go into play. In clinical trials, there are case report forms, which had to be designed prior to starting the trial. There are microbiological case report forms that I have to work closely with data management on to get to include the correct gram stain and susceptibility testing. But I also need a central lab. Each local hospital will run their own susceptibility testing, and then they're expected to send their organisms to our central lab, who will do expanded testing and create a database for us. All of that needs to be coordinated and regulated, and then the database needs to get regular quality control done on it. So we get monthly transfers. You look at it to make sure that all patients have isolates, that nothing was left at the local lab, that now needs to be resent to the central lab. Also make sure that the interpretations for susceptibility are all correct, and all of that goes into QCing it on an ongoing basis. Database lock, the data gets populated into tables, and a study report is generated. And that will eventually go to the FDA. All of that will include a microbiologist who has to look at the data, understand the data, get the microbiological success, failures, and then after the study report goes to the FDA, then there's additional studies that are ongoing. We now have a fabulous set of characterized isolates with clinical outcome. There are some wonderful microbiological studies that can go on with that. Do you want to determine how many were community-acquired versus hospital-acquired MRSA? Do you want to look at which ones make which toxin? Did that affect outcome? So there's a lot of interesting things that can go on at the end of the clinical trial as well. So with the antibiotic clinical trials, and even the large pharmaceutical companies do need microbiologists on clinical projects such as these. So what do I like about my job? I like interacting with all the different departments I get to interact with. I enjoy people. I enjoy being able to discuss with them different parts of science and really show them my excitement for what I do, and being able to convey microbiology, which for a lot of people is complicated. surprising to, I'm sure, all of us. But to be able to explain it to them and see them understand it has really been a fun part of my job. I'm also involved in a huge range from drug development all the way through commercial availability, and that's fun. It's fun to see an antibiotic get to market and do well in the market and continue to do additional clinical trials and see what we can do with that. There's variety. I don't do the same thing day to day. I get to do a lot of different types of projects. I'm involved in so many different aspects now of the commercial group, and it's been a lot of fun. There's growth potential. I started, as I said, I only did external studies. I've been able to go into do clinical trials now, and I do a lot of other additional things, and I just only see potential. I don't really see any big stumbling blocks that are going to stop me from being able to grow, which has been a lot of fun. Another important thing is I have regular hours. I know when I'm going to work. I know when I'm coming home. And yes, there are times I don't get my work done. And then after dinner's made and dishes are done and everything else that needs to be done at home is done, I do sit down at the computer and work on publications or work on various things I have to work on. But I know what times I'm going to be at work, and I like having regular hours. I also get to attend scientific meetings. It's part of my job to, again, go meet with the key opinion leaders and check on my investigations, and I get to stay in with the academic world, and I like that. And I also still get to publish. There are studies that I start, and I work with vendors and external contract labs, and those I do get to work on and write up. And there's a pile of things that still need to be published, but I'm still able to do that and keep my name out there, which is a lot of fun. So how did I get here? It's not a very complicated path for me. I did my undergraduate studies at Miami University in Oxford, Ohio. I got a bachelor's in microbiology and in chemistry. I did my graduate studies in microbiology and immunology at Albert Einstein and Arturo. He was my advisor. And I studied fungal virulence in an amoeba model of infection. It's obviously not what I'm doing now, but with any of the things that you do, it's really learning how to think and process and get the big scope of how to do the literature searches and how to understand new models and all of that. So it's been incredibly useful. And professionally, this is the company I've worked for. I joined Cubist straight out of graduate school. I did not do a postdoc, and I joined in the clinical microbiology department. So why biotech or pharmaceutical? I don't have a real clear answer on that. I went to graduate school to go into industry. I didn't have an academic goal, so that didn't change. It could have easily changed. I had a great graduate experience, but that was my original focus. And I can tell you one of the reasons I think that was my original focus is my father is a PhD in physics, and he was in industry, and he had never been in academia. So I'd seen that my whole life, and I didn't realize all the intricacy of academia at that time. Also, I like initial stability. In the long run, I think academia is more stable. However, initially, I didn't have to worry about competitive grants and NIH pulling funding and reducing funding, and I didn't have to worry about tenure. So that was another thing that sparked me to stay with my industry ideas. I was also very interested in seeing something get to the clinic. I wanted to see the fruit of everything that all of the microbiologists here have done. I wanted to see the overriding picture there. Truthfully, I had no idea what I was getting into. It was an uneducated entry. I thought that's what I wanted, and I went and I did it, and it's turned out fabulous for me, but I really didn't know what industry was, and I've learned an awful lot. And there was a financial push for it as well. How did I find the job? Well, I hit the pavement. That was it. My husband got moved to Boston, so I knew I was going to look in the greater Boston area. Lucky for me, there's a lot of biotech and pharmaceutical there. So I got to Boston, and I started going to all the different companies' websites that are in the area and checking their departments and if they had microbiology and if they were hiring. And I did networked at meetings, and I also used online search engines. In the long run, the online search engine is how I found this particular job. I used monster.com, and it popped up, and then I followed that one backwards from there. Read the job advertisements in the journals that you're reading just to get interested. There will be postings in there. I did apply for industry postdocs. I also applied for several different microbiology and immunology jobs, and this was the one that I ended up liking the best. So There are a lot of other opportunities in industry, and I would love to be able to discuss additional detail on all of these, but I thought I would just bring them up because this isn't my expertise, but it's things I've learned since I've started in industry. There's obviously drug discovery. This is the lab work that we all need and value. There's drug evaluation. Once the antibiotics have been discovered, it's evaluating them, figuring out the mechanism of action, molecular type work, and all of that. It's all there. Non-clinical development, looking at toxicology, animal work, pharmacology, all of those things are also at most drug companies. Clinical development, that's a lot of the clinical trial work that I discussed. Regulatory affairs, dealing with the FDA, dealing with submissions to the FDA. Publications, there's publication strategy individuals, there's publication directors, and these individuals are involved in making sure there's a good publication stream coming out of a company. They come up with a strategy as to what journals we should try to hit that year and in the next 5 years. There's entire departments at some companies that deal with publication and need scientific writers and scientific help in them. That's another area that's interesting in industry. There's quality assurance and quality control, both looking at documents and confirming data, as well as in manufacturing and doing laboratory practices. Program management, overseeing the broad scope of how a drug is being developed and that all the different teams involved in developing a drug are working together in a cohesive way. Drug information, if physicians call in, this is where they get information from companies sent to them, and there's scientists always needed in those areas. Medical science liaison, individuals that are out there in the field, more of a peer-to-peer type relationship with physicians, explaining the benefits of the drug, explaining the data, explaining the new trial. They're out there doing that, trying to get them to do investigator-initiated human studies. So that's medical science liaison. With a lot of, they can be PhDs, PharmDs, or MDs can go out and do this. There's business development, and I think that you heard some of that already, where you can figure out there's opportunities for in-licensing. Where are you going to find them? You need to read packages, understand what other companies have or what other academic institutions have that you can in-license and start drug developing in your own industry. And then there's a marketing position. And that's it. So if there are any questions, I do want to thank ASM for letting me give this presentation. And if there are any questions, I think we're going to answer them now.